Assalamu alaikum. In this video, we are going to talk about artifacts in blood samples. The previous video was about vacuotainers, where we have seen that you collect the blood, then you transport that blood to the lab, and then in lab, the sample is being processed. So in all of these processes, there are many errors that are being done that will that will cause artifacts in blood sample, leading to the false positive or false negative results. So let's see what are those factors. The first factor upon which sample variation depends upon is like lipemia, hemolysis, or icterus. That the sample you have submitted in the lab or the sample being submitted in the lab has either lipemia or hemolysis or is icteric. Like these are important three things I am going to discuss in detail their detail in next slides. Then there are collection artifacts again that are going to be discussed later, and then there are stain artifacts like this artifacts that appear while staining in the lab. We are going to see those artifacts also in detail in the next slides. Next we have is physiologic variations that are due to species, breed, age, or sex. Like first you should know this is not pathologic, this is physiologic. Like some hormones are going to be higher in one sex and while lower in other sex. Same as like calcium, that is going to be in a different normal range in a child, in a young infant animal that is growing, and versus an old age animal that is not growing. So there are like those physiologic variations. Then there are iatrogenic changes, artifacts. These are due to the pre-medications an animal is on, and that is why it is very important that you should have history that this blood sample is from XYZ animal that was on XYZ medication. So that is important because and many drugs have like effects on blood chemistry panels. For example, if you talk about phenobarbital, it increases the liver enzymes like ALT, and we will talk in detail about ALT in a separate video when we will see the liver panel test. But you should remember that ALT is an enzyme. If it is increased in the biochemistry, it reflects liver injury. So if the animal is pre-medicated on phenobarbital and you are saying ALT, you might be like, oh, this animal is having liver injury. But no, this animal is not having liver injury. It has phenobarbital as a pre-medication, so it's normal. His liver is normal. So you have to, in order to reconfirm liver injury, you have to check other enzymes. What are those? We will see their detail in liver testing. But for now, you should know that the drugs have important effect in changing the or causing the sample variations. Then there are pathophysiologic changes, like diet can change the samples. Like diet can change pH of the blood. So you might, if you don't have any like previous history of what animal is being fed, you might say check the like blood chemistry, and you might falsely identify the patient being having acidosis or like alkalosis in the blood. But no, that variations might be caused by the diet. So disease also causes changes. You should know that, and that is our goal, to identify the disease. That's our main goal, uh, to see the changes that are caused by a particular disease. Then there are storage sample artifacts. These are caused by the fact that you have received a sample today that is fresh, but you decide for any reason, you decide to perform tests, panel tests on that sample 2 or 3 days later. So 2 or 3 days later, you are going to have severe changes in those sample. Those are changes are going to be artifacts, not those are going to be false results. But what are these? Number one is hypoglycemia. Hypoglycemia is basically caused by the fact that in a stored sample, the RBCs are going to consume the glucose that is present in that serum of that blood. So when you will check that blood sample after 2 to 3 days, the glucose is going to be decreased, and you will say that this animal is having hypoglycemia. But in actual, that animal is not having hypoglycemia. It's your fault. Next thing is hyperkalemia. If you have a stored blood sample, the RBCs leach out the potassium from themselves. Like from the RBC, the potassium will leach out into the sample. So automatically you are going to have increased potassium, that is hyperkalemia. It is very important fact in horses, especially horses are like most affected. Horses blood samples are most affected by this potassium leaching out. So the level of potassium leaching you are going to see in horses, you are not going to see in any other species. But the other species that are also affected are dogs, especially the breed is mentioned, sheep, cow, again Holstein breed is mostly affected, and the camels. Next thing, our next artifact in storage sample is hemolysis, like hemolysis and cell shape change. Like the blood sample, if you have a stored blood sample, hemolysis will happen in it, and the cells will change their shape, that will affect the test. So here is a guideline for you, that if you want to minimize these artifacts, and you also want to store the sample, like you have a sample collected today and the lab is like in some other country or in some other city, and you have like there is no possible solution that this sample should be sent to that lab within that day. So what you should do to avoid these changes, minimize these changes, is to have cold storage, refrigerate or ship the box. So this should be done. Next, in the next slide, we are going to see again storage sample artifact. This is a storage sample artifact. Up till now, in these three things are related to storage sample artifacts related to biochemistry panel. So it was related to biochemistry things. But this is a storage sample artifact in the hematology. There are like two different tests. These are two different things. One thing is one panel is a biochemistry panel, and the other thing is hematology. You will like fully understand their difference when we will start the series after this lecture. The next series is on biochemistry panel, and after that we will inshallah do the series on hematology, so you will see the difference. But this is something related to hematology specifically, that you see these two pictures. This is a smear preparation from a same blood sample. This was freshly prepared, and then the sample was placed for 2 days, and after 2 days this sample was used to make another slide blood smear and then viewed. Now, after 48 hours, the changes were like you can see there are crenations in the RBC membranes. Also, they are named as echinocytes, and also in the WBCs, you can see neutrophils are being, their nucleus is being swelled. So with swelling, their chromatin is now less condensed. So these are changes that are seen in this blood sample. So you should try to make the slides or like run the panel test as soon as possible. As far as hematology is concerned, in a storage sample in RBCs, you are going to see hemolysis, MCV, MCHC change, shape changes. As far as WBCs are concerned, you are going to see toxic changes, especially Dhole bodies. Toxic changes are basically a term that is referred to WBCs, and they indicate 5 changes. We will discuss the toxic changes in hematology lecture, but till now in this lecture you should remember that Dhole bodies are specifically going to be are going to be seen in a storage sample. Then nucleus will swell, granules will change in WBCs. As far as platelets are concerned, you are going to see platelet clumping. So next thing is hemolysis. If you have a blood sample, like you have taken a blood from an animal, and in that body the like inside that animal the blood was either in hemolysis state, or the blood sample got hemolyzed while injecting it into the vacutainer, or if it is a storage sample, or like because of that hemolysis has happened. So doesn't matter the cause of hemolysis, but hemolysis is going to affect the blood sample. It is going to falsely increase some tests, some factors or some things, and it is going to falsely decrease the concentration or values of the other things in the panel test. I'm not going to in the detail of each and everything, like what is albumin, what is fibrinogen, or what is ALT or these enzymes, because we are going to see their detail in the biochemistry test. So but till now you should remember that these things are going to be affected. So as far as hemolysis is concerned, albumin, fibrinogen, total protein, all of those liver enzymes, then iron, magnesium, potassium, phosphate, bicarbonate, ammonia, bun, creatinine, bilirubin, cholesterol, triglycerides, folate, fructosamine, non-esterified fatty acid, total iron binding capacity, then pro-BNP, all of these are going to be falsely increased. And what is going to be falsely decreased? ALKP, amylase, bicarbonate, bile acid, GGT, NIFA, and SDMA. So you can also see that ALKP can be decreased or can be increased. So hemolysis has effect on all of these tests. Next we have is lipemia. Lipemic sample looks like this, like it has cloudy appearance. You should remember that with lipemia there is excess fat in sample, and that fat interferes with light scatter that is used in some testing. So all of those testing are going to be affected. It falsely increases albumin, fibrinogen, total protein, these liver enzymes. This is muscle enzyme, then ammonia, bile acid, cholesterol, calcium, TIBC, and troponin. It falsely decreases bicarbonate, bun, chloride, sodium, potassium, and LDH. Next we have is ecteric blood sample. Ecteric blood sample means that your animal is having jaundice, and because of that there is like bilirubin is being increased in the body. So because of that your sample gets this like bright yellow. This is basically a centrifuge blood sample. So hematocrit is down here and this is serum, and you can see serum is yellowish. So ecteric blood sample falsely decrease. Basically I have this arrow should be like downward, sorry guys. And because it is, this falsely decreases these things, and it falsely increases, so it arrow should be like this. It falsely increases ammonia, AST, CK, bun, bilirubin, calcium, and it also interferes with bile acid, cortisol, fructosamine, and LDH. Like it not specifically increases or decreases it, but it interferes with these tests. So you should remember that these values are going to be affected based on what type of a sample that is being submitted. Next are collection artifacts. First thing is difficult venipuncture. Like you are a beginner, you are a student, and you are collecting the blood sample, and you do not have a good skill. So what you are doing is inserting and reinserting the needle in the blood vessel, and because of that, platelets will clumps. And if you are going to, or if the lab is going to check the platelet count, it is definitely going to be lower because platelets are already being clumped. There will be micro clots in the sample, there is going to be hemolysis in the sample. So you do have good skills. Then low sample volume, like we have already talked in the previous video that EDTA is hypotonic. And if a tube says you should administer 5 ml sample in that EDTA tube, and you administer 1 ml because your skills are bad and you manage to just collect 1 ml blood, then it is going to cause the shrinkage and lysis of the RBCs, and that will decrease the false, there will be false decrease in MCV, false increase in MCHC values. If there is rough handling, like you have like hemolysis, rough handling will cause hemolysis because of the forced insertion of the blood through syringe into that tube, or because you have like done very extra shaking vigorously. Because of that there will be like hemolysis. Then you have used wrong tube, like each color top tube has separate indications that we have discussed in the previous video. So but you had you like you are using calcium, like you are using blue top tube for calcium analysis, but you know, or you are using purple top tubes for calcium analysis, but you know that calcium is going to be chelated by EDTA, and you are going to have false results. So you should know what tube you are using. Then last we have is stain artifacts. Like this artifact is like in specifically related to the hematology, that you are preparing a slide and you're going to have different artifacts. Number one is water artifact, because of presence of excessive water in the fixative, or and because of that you are going to have irregular punched out areas in the RBCs. Like you can see this picture, these all areas are like just like punch out, just like you punch a paper and a hole gets developed in that paper. Similarly it looks like punched out areas in the RBCs. The second is stain precipitate, use of that happens because of age, staining in the solutions, inadequate rinsing of the slides following application of the stain, like you haven't rinsed your slide properly after staining. Then stain precipitates, you can see this is a stain precipitate, and precipitate mimics the bacteria. It looks like bacteria, including cocci or mycoplasma. But you, how to differentiate between this? You can differentiate differentiate the stain from bacteria. That stain is going to be precipitated, and it is going to be purple in color, whereas bacteria is going to pick the blue color with the right stain. So that's how you are going to differentiate this. So these are two most important stain artifacts that are seen normally because of the errors that are mentioned over here. So that was all about artifacts in the blood sample. I hope you guys have understood this. Do subscribe and like. Thank you.